As we move into the technical presentation, and before we get proper into the session one, which would look at methods, we have a featured technical presentation from one of the lead project partners to kind of set broad tone for what we'll be talking about throughout the other sessions. And so I am pleased to invite to the podium again Dr. Laurence Maurice, Director of Research at IRD, and she'll be speaking broadly on the impacts on the mercury biogeochemical cycle and mining. Laurence?
Yeah, that's okay. Thank you very much, Calvin, for introducing my presentation. So that's okay. You can hear me? Yeah. So today I will give you some keys to understand the complexity of the mercury biogeochemical cycle and how the AGM, artisanal small-scale gold mining, is impacting this cycle, and what are the impacts on the environment, but also on the human health. So I will first give you some numbers at the global scale, and later I will zoom on different case study in. Is that okay? On different case study in South America. I will also present you some examples of experiences of empowerment of local population we had in Brazil and in French Guyana. So, no, but what are you doing? So first, everybody, I suppose, knows what is mercury. Mercury is a metal, white of the right color. Silver, it's called quicksilver. And the symbol, the chemical symbol of mercury is Hg, because the Greek name is hydrargyrum. And you can find it in different chemical forms. You can find mercury in vapor form in the atmosphere. You can find it in a dissolved form in inorganic and organic species. And I will insist a lot on the organic species, which is methyl mercury, which is the neurotoxic, which is the most dangerous form for a human and for the living life. And we can also find it solved on particles, on sediment, for example. So the mercury is emitted all over the world. We say that there is every 2 years there is a very big conference dedicated only on mercury, and the title of the conference is Mercury as a Global Pollutant. Mercury is a global pollutant. You can find it everywhere, in the air, in the atmosphere, in water, in fresh water, in marine water. You can see it from the Arctic to the Antarctic, and also, of course, around the tropics. It's a real global pollutant, and it has been emitted since a long time ago. So this is the presentation of the history of the emission all over the world, and the first boom, the first rush was in, of course, during the Inca period, but also in the 19th century due to the American gold and silver rush. And later, for the World War, first and second, you had a decrease of mercury emission. And from the 19th centuries and the 20th century and right now, the first source of emission in the environment are the coal plants. So if we present the mercury emission all over the world right now, the first country that emitted mercury in the environment are China, mainly China due to the coal plant emission. They were building like one coal plant per week in a certain period. So it's the first emitter. And the second country is South America here, mainly due to the aids to the gold mining industry. And later you have Africa also. There is a lot of gold mining activities in Africa that impact the global emission. You can find all this data in the last report of the UNEP. Regarding the part of South America in the emission of mercury, here it's South America. I'm sorry, it is quite small, but the screen is quite small. So here you can see here it's Asia, source of Asia emission, and here is South America mercury emission. And in yellow, you see the part of the emission due to the gold mining activities. So of course, in South America, you can calculate it represents 53% of mercury released in the environment, in the atmosphere. So IGM is responsible of almost 40% of global mercury emission all over the world. It's the first source of mercury in rivers, which has been estimated recently to almost 900 tons per year. And South America is responsible of more than 50% of mercury release, only by artisanal and small-scale gold mining. And of course, it's not only the mercury emitted by the gold mining activities, but also emitted by the deforestation related to human activities. The deforestation has increased in the Amazon rainforest during the period between 1985 to 2020. It has grown of sixfold all over the Amazon rainforest. And I know that WWF is working on this issue all over in the Guyana plateau. So, of course, the AGM is also responsible. It's a large contributor of the gold supply all over the world. And the gold is mainly used in jewelry, retirement fund, electronics, and national treasures. It employs till 19 million of people all over the world, so mainly in South America and in Africa. It's a high contributor to the employment, and also, unfortunately, it contributes to finance conflicts, mainly in Africa, and also facilitate money laundering. And I won't speak about all the prostitution and guns and arms laundering and so on. So it's a global issue which has an important impact on not only on the environment and human health, but also on social and economical issue. So here is the map of the mercury emission all over the world. So you see the most important mercury emission in the world is here in China, later in South America, in gold mining countries, and in Africa. So the natural sources of mercury, there are of course natural sources of mercury in the environment. You have the cinnabar is the main rock which contains a lot of mercury, and it has been exploited in Spain and also in China. You have several mercury mining in this country. The other source is the rock lixiviation, soil erosion, and also the emission by volcanoes. Volcanoes are the first natural sources of mercury emission in the world, and we have estimated the global natural emission to 600 tons per year. Regarding the anthropogenic sources, the sources due to human activities, one of the most important, which is the issue of this conference, is our mining activities. Of course, mercury mining, gold and silver. As I explained to you, the energy production by coal combustion, coal plants. It's also used in the electrical industry, in mercury vapor lamps, in fluorescent tubes, and even in low energy lamps. It contains an important concentration of vapor mercury, so be careful. You don't have to break lamps in your house or everywhere, because it may emit mercury vapor in your environment. It's also used in the chemical industry, chlorine, caustic soda production. It was used, but now It's forbidden in thermometers, barometers, thanks to the Minamata Convention. In dentistry also, there's a large action plan to reduce and to stop the use of mercury in the dentistry. It's used in jewelry too, and of course agricultural practices also impact the mercury cycle and increase the mercury fluxes in the atmosphere due to the biomass burning and also to the deforestation. So you have a global budget. It's really small. I'm sorry, the screen is small, but it's just to show you. This is whoops. This is the geogenic emission, which are 500 tons per year, well, very, very small, from terrestrial activity, from volcanic activity, and in marine activities. And here is the 100% anthropogenic emission, and in yellow you have the emission of mercury after reprocessing of natural and anthropogenic sources. I won't insist so much. But what I wanted to explain to you now is the mercury cycle in a mining area, in a AGM area. So, for example, you have, of course, it depends on the country. In Guyana, you don't have volcanoes, but I was working in Bolivia, Ecuador, Peru, and so on. So, of course, you have mercury emitted naturally by volcanoes, but you also have mercury emitted during the amalgam burning in the atmosphere, and the mercury vapor in the atmosphere is quite stable. It can travel on hundreds and hundreds of kilometers in a dry atmosphere, and when the atmosphere is becoming wet, this mercury vapor is reduced and can precipitate with the rainfall and can fall on the soil, on the rivers, in the lakes, and so on, and re-enter the hydro system. And in the lakes, in the rivers, you have several processes that modify the speciation of these elements. And one important point I would like to insist is that the mercury which you may find and measure in soil and in the atmosphere in soil is trapped with organic matter, is trapped in the soil and is quite stable. Once it enters into the water, into lakes, into rivers, you have different processes that change its speciation, the speciation of the mercury, and it can be methylated. That is to say that you may find some bacteria, sulfatoreductor bacteria, that transform the inorganic mercury coming from the soil erosion, coming from the atmosphere, into the organic mercury form. And the methyl mercury is the organic mercury form, which is highly, highly toxic. And this methylation process occurs mainly in the roots of aquatic plants and also in sediments. Yeah? So if you want, I can explain later and I can answer your question. But it's an important process because it's the organic mercury which may enter later in the trophic chain. And of course, you have also two processes that increase the concentration of mercury in the trophic chain, the bioaccumulation process and biomagnification process. And later, the incidence of mercury in the human health, of course, depends on the chemical form of exposure between inorganic mercury and organic mercury. The inorganic mercury is inhalated, the mercury vapor is inhaled by gold miners and gold sailors. Gold miners also use liquid mercury, which can enter into the by dermal exposure, but it's quite few. It's quite small. The most important exposure is via inhalation, And the second exposure pathway is via the diet. As I explained to you, the mercury is bioaccumulated and biomagnified in the trophic chain. So you can measure very high concentration of mercury in fish, in piscivorous fishes, which can be consumed by local people. And this organic form is the most important exposure pathway. So the toxicity of liquid mercury on the human air. So you have three types of exposure pathway, as I explained: inhalation, ingestion. I met some gold miners who told me that sometimes some of them ingested the amalgam, the combination of mercury and gold, to avoid to declare the production they found. So sometimes some gold miners may also absorb liquid mercury. And the third exposure wear is dermal contact. And the effects are, of course, important, but depends on the exposure level. So you may have neurological disorder, muscular disorder, kidney, and so on. And you also may have, when you are highly exposed, you also may have hallucination, psychosis, gingivitis, trembling, and so on. So for this exposure, for the human exposure to inorganic mercury, the best biomarker is the urine, because the mercury is entering in the body and is excreted via urine. So urine is the best biomarker to measure the gold mining exposure. And you have thresholds that can be overpassed depending on the places. Regarding the toxicity of organic mercury, it's mainly due, as I explained, to the diet by eating fish. And the best biomarker for studying the organic exposure is the human hair. And we can observe different incidences, different impacts on hearing, visual. You have a reduction of the vision, renal functioning, coordination of the members of arms and legs. You have also, you may have, it also affects the immunity. And when people are highly exposed and for a long time period, it can also affect the behavior. You have a reduction of the intellectual quotient for children who are exposed till during the pregnancy. It affects the memory, the attention, cerebral, till coma. So the absorption of mercury by the human body is quite, it's an important issue, because the organic mercury, presents particular affinities for the brain. The human brain is, and the ratio we measure between the mercury in the human brain and the mercury in blood is around 6. So the organic mercury is also concentrating, is also trapped into the brain. So that explains the incidence on the neurological development of people. The exposure of people to methyl mercury depends on the doses and on the time of exposure. People were exposed since the pregnancy, since they are baby, it affects the neurological system, and its irreversible effects. Of course, the population with major risk are the regular fish consumer, and the neurological consequences are irreversible. So in the aquatic food chain, you have a high increasing of mercury concentration between the water. In the water, you don't have dissolved mercury, and Loic will present the results we have in Guyana. But what is happening is that both different processes, methylation, bioaccumulation, and biomagnification, affect affect the mercury concentration, and you can have a differences between the water and the muscle of piscivorous fish of 1 million. So you have a biomagnification of the mercury 1 million-fold between water and fish. So the factor that controls the mercury in fish species are the trophic level. Of course, fish which are eating fish, of course, are more concentrated than omnivorous fishes. The age and the size, the growth rate also affect the mercury concentration. The hydrologic period, if the species is sedentary or migratory, and the place where the fish are living, because we observe that in lakes, in floodplain lakes, in reservoir, we observe a high methylation rates, which of course increase the mercury concentration in the fish. There are a lot of documentation on the fish species who are highly concentrated in mercury. We did several studies in Ecuador, Bolivia, and Brazil. So you have a large contribution, you have a large bibliography on this issue. In French Guyana, we also observed, because in general you will find information on mussel, because people mainly eat mussel of the fish. But in some places, and in Bolivia, I was working with indigenous population, we were also eating the liver of the fish. And what is happening is that in the liver of the fish, you have in general higher concentration of mercury than in the mussel. Why? Because in the liver you have a detoxification process that occurs in fish, but also in human body. And we also measured a demethylation process. Your body to detoxify from heavy metals, from toxic metals, it occurs different processes that try to detoxify, and one of the detoxification processes is the demethylation that occurs in the liver. So in the liver, the concentration can be higher than in the muscle. And also, we have a biologist colleague that analyzed the effect on the fish, on the muscle, and also on the liver, for example. And these are fish we were caught in a pristine place, and this in a mining area, and we observed an hepatitis of this liver cell in highly exposed fish species. They also did photography of muscle cells in three different areas, in pristine area and in gold mining area, and they also observed an affection of these cells, and they also observed a difference in the way of swimming of the fish and so on. So this is for piscivorous fish, which are highly exposed to mercury. But also, so I was speaking of the Amazon and of the fish species that we can find in the Amazon basin, but also in the ocean, the piscivorous fish are also highly exposed to mercury, and specifically tuna. So the tuna fin we can measure high concentration of mercury in tuna can. And for example, in Canada, on the tuna can, you have an advice for pregnant women, indicating that be careful, you may not eat. You may eat just 1 can per month if you are pregnant. So there are also a lot of bibliography. There are a lot of literature on the mercury in tuna and also in marine fish. On the Amazonian community also, we have a lot of data. I first worked in Bolivia, and the concentration, the highest concentration I measure was of 35 microgram per gram, while the rule limit is 10. And colleagues observed the first weeks at 5. So it's we can find in Amazonian people very high exposure, and these people are living in the Amazonian forest in area without any gold mining activities or other human activities. So and the limit we propose of 0.5 in fish, it's for commercialization. But what we observe is that for people whose diet is based on fish, it's quite difficult because in the Amazon basin and even along the rivers in South America, a lot of people, the main source of protein are the fish. So there are several maps of mercury of human exposure over this basin. We published in 2009, and now WWF in Brazil proposed also a mapping of the mercury exposure of human and also of fish, and we are working on the actualization of this database. In French Guyana also, we observed that people. So we were working along the river between Brazil and French Guyana border in Camopi, which is the mining area, and Troisseau, which is a pristine region. And we observed that Amerindian people living in the pristine area are more exposed to mercury than the same community living in the mining area. So, of course, it was a bit different than we could expect it. So we developed a tool, a geochemical tool. We are now using a stable isotope, and I think that a colleague of University of Guyana will present some data. So with the stable isotope, we can also measure the, well, so I will go quickly. So we did the analysis in the sediments. And this is the fractionation of mercury in sediment. Here is the liquid mercury end member, and here is the sediment of the pristine area end member. And all the points of the sediment results are in this line. And with a small formula, we can estimate the part of AGM in this drainage basin. So the study has been published recently, and active AGM can increase by 78% the mercury concentration in sediments in active rivers. We also measured this fractionation on human hair, on water, on sediment, on fish, and also on human hair. And it was interesting because between the pristine area, Troisseau, and the mining area, the women present exactly the same. The main source of mercury is coming from the freshwater fish, while the men, we observe a difference in the fractionation that indicate that they have another source of mercury, and with the questionnaire we did, they were also mixing their diet with can and sometimes with tuna cans. So it's interesting to see that isotope can also help you to identify the different sources of mercury in the human diet. So we also worked on the empowerment of local population. There are several projects in Brazil and in French Guyana to help people to reduce their exposure, to limit, of course, the incidences. So there are a lot of data on that. And the problem with the mercury exposure is that it's invisible. You don't see in your body if you are highly exposed or not. And so we, in Brazil, colleagues from Quebec proposed some comics to explain all the processes that could impact the mercury cycle. And also they proposed a poster with fish species that people could eat and not eat. Of course, it was essential not to create a psychosis about eating fish, and but they also explained to them what are the impacts on the vision, on pregnancy, and so on. So after this information campaign, they observed decreasing of almost 40% of the mercury exposure in the population of the Tapajos River. We did that, so I will go quickly. We did that also in French Guyana in 2014. We organize the biomonitoring on the main river of French Guyana, and we also proposed different posters of fish species that people could eat daily, and fish species that women who are pregnant and even kids could avoid or eat just once per month. So this poster has been realized after large analysis, a large amount of mercury analysis in fish, more than 6,000 during 10 years. And we also propose the same advices than in Brazil. And we also observe a decreasing in 10 years of the mercury human exposure. So now we are moving from academic research to co-construction of adapted and sustainable solution with local people, with gold miners, with stakeholders, to co-construct with the population solution, allowing to adapt and reduce the environmental and health impact, but of course respecting the traditional knowledge and culture. So I'm done. Thank you for your attention.
Thank you very much, Laurence. And thank you for cooperating with me. So we're going to go into those sessions. You'll note, based on your agenda, that we will have a series of presentations that are somewhat related, and then we'll take the panel of presenters to have the questions and answers. I'm sure you may have questions in relation to Laurence's presentation, but she will be in the panel that comes up after session 1. So with that, I'll hand over to my good friend Patrick Ketwaru, and he is going to be the chair for the first session. Patrick.
Good morning, everyone, and welcome. This is going to be the first session of, you could say, more technical stuff. And I think we had quite a good start by Laurence's presentation. So that gave us quite a wide overview of the issues. And what you'd find is that these presentations, these technical presentations, would actually now move into various aspects of that. So let's get started. So I'll ask Dr. Lachmansingh to come and do his presentation, which is based on the methods proposed by the project as standard for monitoring mercury in environmental and biological samples. And there is need for standardization, and those are some of the things I hope we'll be able to discuss in the panel.
Good morning, everybody. So I'm Loic, working with Laurence at IRD in France. So I'm going to present the set of methods we used during the project, the Mercury phase-out project, and when we sampled all the environmental compartment and biological sample. So the idea is to show the protocol we use in our lab in Toulouse, and how it could be used to monitor the environmental level of mercury in aquatic system, and also the level of exposure of people living in riparian area. Okay. I use that to... okay, perfect. So we have type of sample versus type of matrices to work with the different kind of analysis and sampling we can do in aquatic system. So first, biological sample. So it will be human air to assess the level of exposure of people. It's a good biomarker for methyl mercury, the form that Laurence presented as the most dangerous for human beings. And this exposure is done by fish consumption, so we need to sample fish tissue to assess the level of mercury in those animals. There is also environmental samples like water, fresh water from river, suspended particles which are in suspension in rivers, soil, sediment, and litter and leaves. Then the matrices that comes with that are either liquid or solid. So the analysis will depend on the kind of matrix we have. First, before to do any analysis, we need to sample the sample. When we sample water and sediment, when you have nice water like that, like this one, you see it's like a bit of mud. We use some PTFE bottles to collect the sample from the pond, from the river anyway, and we have to filtrate it in a filtration tank, where on the top you have the not clean water, let's say like that, and then the filtrate, and in the middle we use Teflon filters to recover the suspended particle, which we will analyze for mercury in suspended matter. When the filtration is done, we stabilize the sample in Teflon bottle, the filtrate, with 0.4 HCl in volume, and we store it at 4 degrees to avoid any mercury loss or stuff like that. And we keep the filters in a Petri slide box, small box where the filter is isolated from the atmosphere, and we dry it under laminar flow hood. This work is quite not difficult, but we have to be careful when we work on those samples because there is few mercury in water, really few, and possibility of contamination from anywhere around could be really not dangerous, but could happen really quickly and really easily. So we need to work in quite clean environment, and we need to work with clean acids. Like in our lab, the HCl we use is either a trace metal grade or by BD style. All tanks and everything are washed with 10% HCl before to work, and we on the field it's not possible to wash everything with HCl, of course. So in between samples, we wash it with liquid water or ultra pure water that we carry with us on the field. When we sample soil, sediment, and tailings, it's quite easy. This one you don't risk any contamination from cross-contamination from the environment. But what is really tricky is that we need to sample at the right position, especially for sediment, because you have sediment transport in rivers, so you cannot sample everywhere. Sometimes, otherwise, you will just find sand and you cannot do anything with that, or you won't target the right position, or you will have the soil from riverbanks, for instance, coming to the river, and this is not the one you want to have to understand what is the level of mercury in your system. So basically, we sample everything with Ekman bench, which allows us to go in the middle of the river and sample the bottom sediment. We use Odger for soil, and then we store it in Ziploc bags, and we work with and we dry it before to go to the lab. When we come back, when we arrive in the lab, we dry it at 35 degrees, not too hot to avoid any loss of mercury, and we homogenize everything with sieving first, and then we crush it for analysis. Fish. When you collect fish, you need, for mercury and understanding, you need to have length and weight. It's on scale. Then we take a small part of the flesh with a clean knife, and we freeze it before to go to the lab, and then we freeze dry it and crush it before analysis. Human hair. So you see, Laurence is collecting some hair from a past mission. We take strength from the back of the skull, like 3 or more. Depends on the size of the strength. For men, it's more than 3, and for women, it's easy to have enough. And we store it in bags, and we chopped it before to analyze it. So now we come to the part of analysis, which is where it's more interesting, I guess. Liquid. So for us, we analyze only water in a liquid way, and also we mineralize suspended particulate matter from filters. So the preparation is quite easy for waters. We just oxidize mercury organic complexes with bore chloride, which allow us to have only mercury 2 in the dissolved water. For suspended particle, we do a digestion on hot plate in a clean Servilex. We dilute each extract, and we also use oxidation with brom chloride to have only mercury 2 in the solution. Why mercury 2? Because when you analyze mercury, we do it with a gold trap called vapor at atomic fluorescence spectrometer, which means that the first part is reducing mercury 2 in mercury 0. Mercury 0 is a gas, and it goes to the gold trap. The gold trap helps us we do the same as the miner, but in a reverse way, let's say like that. It helps us to pre-concentrate the sample. Then we burn the gold trap, and we have a classic atomic fluorescence detection. And this helps us to reach low level of mercury concentration. We go to until 0.5 nanogram per liter, which is 0.5 ppt. It's quite low, and we can go really, really low in concentration. Okay. Solid, we use either a direct mercury analyzer or an advanced mercury analyzer, the technique is the same. Basically, you burn your sample at 800 degrees. The mercury is released as a gas. We have, how do you call that comment Laurence, le truc après le détecteur, c'est quoi? On dit comment en anglais? C'est sur le DMA. No, okay, no way. I forget the name in English, sorry. Yeah, but basically the sample is cleaned through active carbon to avoid any interferences, and then you have a classic fluorescence analysis. And then this is quite easy because you don't need further preparation for the sample. When you take a fish, a solid, anyway, a sediment or I don't know, sediment and a human hair, you just put it in some boats here and you do the analysis. So it's quite easy. It just has to be crushed to be really well homogenized. So in all the case and all the technique I presented, we have to do calibration, of course, and quality control. So all our analyses are externally calibrated. The calibration standards are done in the same matrices of the sample. So when it's a sediment, we use a sediment as a standard. When it's a water, we use a water. And also, for instance, when I analyze filters, I use the same matrix as the filters. We consider two types of blanks: the analytical one, all the analytical reagents, and the blank has followed the same procedure as the sample to be sure that we don't have any cross-contamination during the process. There is also, of course, reagent blanks to be sure that the acids are in good quality. When we work in mercury, the quality of the acid is really important because it's the first way of contaminating your sample, especially because a pure chlorhydric acid, if it's not stored really well, for instance, will trap the mercury from the atmosphere, and you will have a polluted acid. And if you use it to do your analysis, at the end you will pollute your sample. We calculate detection limits, which is the derivation of we do it with 12 blanks. Most of the time it's analytical blanks, like that we know what is the quality level of our analysis, and the detection limit is 3 times the mean of the blanks, and the quantification limit is 8 times the mean of the blanks. All samples are analyzed in triplicates. Like that, we have the variation of the concentration. And we use certified reference material to control 3 parameters, accuracy, yield, and reproducibility. For mineralization, also, we use a certified reference material to check the yield of the protocol. That we include one in the sample to be sure that we find the right concentration at the end of the process. So the accuracy is basically the standard deviation of your measured certified reference over the mean of the measured concentration. The reproducibility is the average, the mean of the concentration measured over the concentration certified, -1. And the yield is the mean measured concentration over the certified concentration multiplied by 100 to have a percentage. The yield is the one we use the most to control the quality of the data, and also the evolution of the runs, because sometimes we analyze 100, 200 samples in one day, so we check also the deviation of the signal during analysis and so on. So here is a list of the standard reference we use for mercury analysis. Every time we choose them in function of the type of matrices we analyze and the expected concentration range. So for sediment, we use, of course, estuary sediment, sometimes coal when we want to go in low concentration. We have polluted soil also that could be used when it's a high concentration in mercury for tailings, for instance. And when it's for human air, we have also some reference material. And for fish, we use lobster hepatopancreasis and also muscle tissues to check the quality of the measurement. So yeah, that was my last slide. So basically it summarizes how we process all the samples we collect from the field and what we did here during the field mission. And all this protocol was used to have the data I will present a bit later during the day for the project. Thank you.
Thank you, Laurence. That was quite interesting, and we actually do some of this work right here at the university. And it would be, I guess, if anyone is interested, tomorrow we have a few students in our final year who would actually be going through the coal vapor analysis. Tomorrow morning we'll be doing that. Thank you. We go on to Elford, Miss Elford, Liverpool. Doctor, yes? He's there, he's going there. And he will be looking at his topic. Thank you.
Thank you, Chair. Good morning. All protocols observed. Today I'll be complementing some of what Dr. Luick just mentioned in the context of what we did in the middle and lower Mazaruni sections in Guyana, where we try to integrate stable isotope analysis to reconstruct food web and look at food web ecology in fishes in the Mazaruni River. This is a collaborative initiative with Dr. Montana and Dr. Taporn and other colleagues from the US. And so the objective really was to, the objective really was to Yeah, the objective really was to see how we can make interconnections with test. Yeah. Check. Yeah. So the idea is to apply stable isotope, as I mentioned, to model the movement of mercury through the aquatic system in the context of food web ecology. So we set out to explore a number of methods and a number of initiatives in order to gather data to try decipher what's going on in terms of food web ecology in the middle Mazaruni sections of Guyana. So stable isotope in general is a unique tool that is used to provide insights into various aspects of ecological processes, trophic dynamics, and more importantly, it also can be a useful tool in providing insights into pollutant pathways. And it's also crucial in understanding and managing, for example, freshwater ecosystems. I know my two colleagues earlier presented on the mercury isotopes, and Loic went into more details with regards to the laboratory assays with regards to testing for mercury. So mercury isotopes are usually used as a tracer of pollution, just like any other heavy metal within the ecosystem. And it is very crucial in terms of our understanding of how mercury moves through the environment. Carbon isotope is an important measure of primary production within an ecosystem. And so it gives us an insight in terms of energy sources within the ecosystem. So it kind of gives us an idea of where our sources of energies are coming from. What do we eat, right? So you can look at an organism, run a carbon isotope analysis, and you can tell the sources of where they're getting their food from. In more complex models, you can actually calculate the percentage of the various sources of your food resources using carbon isotopes. Nitrogen isotopes give an idea of your trophic position within the ecosystem. So whether you're an herbivore, whether you're an omnivore or a carnivore, or whether you're feeding at the top of the food chain. So it's also an important tool in terms of deciphering what's going on in terms of vertical movement, for example, of mercury within the ecosystem. So by combining these three isotopic tools, we can have a more comprehensive understanding of the movement of mercury within various pathways. So here is an example. Here we have a simple biplot model of some basal food resources, and when I say basal food resources, these are usually the photosynthetic arm within the ecosystem. They're capable of making and producing their own foods. We have invertebrates, so many of these invertebrates would feed on the producers within those ecosystems. And then in turn, we may have fishes that feed on these invertebrates or may feed on the basal food resources themselves. And in turn, that creates a chain, and it can go on to be more complex of a nature in terms of a food web. Then you have humans going and eat those fishes, and they may probably go more above those black triangles that we're seeing there. But what basically happens is that the closer you are to a particular source in terms of the carbon isotopes, your source of energy usually comes from that source. So for example, most of the fishes that are in the black triangle there and the invertebrates, they usually get their food sources from the extreme right of the graph. Those are the periphytons. So you can see a kind of vertical movement going up from the basal periphyton, invertebrates feeding on those, and then the fishes feeding on those invertebrates. Within that complex matrix of the fishes, you have omnivores, you have carnivores, and you have piscivores. So the piscivores are usually the top predators within the ecosystems, and they would usually feed on other fishes. And so they are usually the ones that have high evidence of mercury by accumulation within those systems. So what we did in our study is that we carefully integrate a number of techniques, starting from site selection processes. What we tried to do, we tried to compare data that was collected between sampling sites that were mining impacted versus sites that were not so mining impacted within the stretches of those rivers and their tributaries. After careful selection of mining and non-mining impacted sites, what we went on to do is to try break down those sites into further subcategories of macro habitats, whether they're riffles, whether they're runs, whether they're pools, so that we can have a varying subcategories of habitat or variability in terms of habitat types that we sample. So we're not only sampling pool, we're sampling where we have rapids and riffles, we are also sampling where we have flow. These different habitats have different productivity levels and so on, which I won't go too much into. But in addition to that, we also want to sample sites using a number of different techniques, and these techniques that we use must be statistically comparable across sites. So one of the important components in terms of our sampling is we do a diversification in terms of our sampling techniques. When it comes to, for example, fish sampling, we use a combination of gill nets, we use dip nets, we use kick netting, sail netting, and in some cases we use traditional fish poisons from our Amerindian brethren, and we also would procure fish samples from, the regular cart during our field surveys. So it maximizes our sampling, it maximizes the diversity in terms of fish sizes and also the different types of trophic positions. So we must be able to capture fish samples that span herbivores right up to the tertiary consumers in our sampling strategy. So for us to build a better understanding of how much mercuries are being positioned at each level and how it's being bioaccumulated. Invertebrate sampling usually use a number of other techniques. Mainly these are say netting and kick netting when we do our samplings as well. And so we have made that a standard part of our procedures because they're also part of the consumers, the primary consumers within those ecosystems. Fish body size in terms of total and standard length are usually a form of measurement, and it's important as a measurement of our bioaccumulation factor and weight. We usually measure those in and the weight we take in grams for all specimens that we're sampling within that survey site. Muscle tissues are usually the primary focus in our case. We usually take a dorsal piece of muscle tissue from the fishes, and we preserve them for mercury, and we preserve them for isotopes. For isotopes, we usually put them in a sterilized Ziploc bag, and we add salt. Common non-iodized table salt seems to be the best preservative technique for preserving stable isotope samples so that we can take them for further laboratory analysis. For mercury samples, we usually store them on ice or in liquid nitrogen in sterilized vials or sterilized Ziploc bags as well. Earlier, my colleague pointed out there are other parts that are also retain high levels of mercury, like the liver, and so that may be another technique that we can integrate other than collecting standard fish muscle tissues for sampling. In terms of primary producers and other basal food resources, we usually try to take for each site, all the possible sources of food for the various fishes that we're sampling. So we will collect moss and algae, we'll collect fruits, leaf litter, and any other surrounding organic material or detritus that are present within that habitat we're sampling as part of a more comprehensive technique in terms of sampling for stable isotope and to have a better understanding of what these fishes are feeding on when we're building our models. In addition to that, basal resources and other samples, we also take water samples and sediment samples as outlined by Dr. Lewick earlier. It is also important to note that with building these food web models, it's important to take reference and baseline material. So we must have reference material in terms of water sample, atmospheric samples, sediment soil samples for especially mercury isotope if we want to trace, for example, the source as outlined earlier. We also do take a number of environmental parameters, including pH, temperature, dissolved oxygen, turbidity, which is indicator of mining pollution in many of our freshwater streams. And one of the more extensive components in our sampling is habitat description or habitat characterization, where we look at all the different characteristics that are either substrate based within the channels or streams, and we look at surrounding riverbank characteristics like percentage trees, percentage shrubs, grass, leaf litter, and so forth, soil type. So we build a more complex data collection sheet when dealing with data collection for modeling mercury. Our samples, when we bring those samples out, we usually prep them and we send them overseas for laboratory analysis using standard procedures as outlined previously. In terms of statistical analysis there are a number of tools that are out there that we can plug all of these data that we collect in, and we can come up with more complex models to give us an insight of or deciphering what is going on in terms of energy flow and mercury contamination within and up the food chain. Here is an example of what we found in our initial survey. In our mining sites and our non-mine sites, we found that most of the fishes in the mine site are more associated with algal, with detritus or benthic algae, and there's some variation in terms of what's going on in terms of non-mine sites. But all in all, in both non-mine and mine sites, we found that mercury is being bioaccumulated in the tertiary consumers, more so the piscivores and the carnivores within those ecosystems. And so this vertical movement of mercury within that system. In addition to that, a use of total mercury and nitrogen isotopes provides an insight in terms of bioaccumulation as well of the food chain. And so it's a clear picture of how the mercury moves up the food chain as well. Many of the fishes that we collect are food fishes, and so if you notice, they are above the WHO guidelines for consumption. And that's the end. Thank you.
Thank you, Alfred, and that is the end of this first session, but I'd like to invite the three presenters who presented just now to go on stage, and we will then the audience, you're free to ask questions. They move the mic up there.
Just if you haven't had a snack, please feel free to quickly as you get here to grab something. The solids are there now.
So do we use a roving mic or what?
We have where's the second mic?
While you're having your snacks, you could go ahead and start. We could start with the questions, right?
Yes. Advise people online, they can indicate in the chat.
Okay. And for those online, you could indicate in the chat that you want to ask a question, or you could type it in, and we'll pass it on to the panelists. So over to you guys. Ask a question. We had some very interesting presentations, and it was quite a wide scope, so feel free to ask the questions.
When you're asking, just so that the rapporteurs can keep recording, just identify yourself, please.
William Wolford, I'm representing the Guyana Gold and Diamond Miners Association. I have just a quick question to understand how it was done, Mr. Liverpool, I think it is. You said Middle Mazruni. Which area you're calling Middle Mazruni? You could give place names. And secondly, do you sample in the main Mazruni or in the creeks that flow into the Mazruni?
Thank you, Mr. Wolford. The Middle Mazruni section that we worked was from Isinuru right down to Bartica. So the middle and the lower Mazruni. Okay. In terms of sampling was done both in the main channel of the Mazruni as well as all the possible tributaries and creeks within that system.
I asked that because I didn't hear you mention seasonal variation. In the wet season, it's horrendous. How did you manage that? Or you didn't sample in the wet season?
We did both wet and dry seasons during that survey. Yeah.
You managed to sample in the wet season?
Yes, we managed to sample in the wet seasons.
All right.
We go where it's wadable and where we could, we can fish within the water without risking, drowning.
Yeah, but what we found in practice is that during the wet season, the fish seem to go into the side creeks.
To the side, yeah.
So you wouldn't...
We use gill nets, kick nets, a number of different combination of techniques to sample those
all right.
fishes.
And then just a quick comment. You mentioned at the last that you send the samples overseas.
Yeah.
Anything working in our favor to get the sampling test work done here?
Yes, because that was a collaborative initiative, the idea was to send the sample overseas initially, but now the university through the chemistry lab has that facilities, and as Mr. Ketwaru pointed out, there's the capacity to run those analysis now.
To you, Laurence. One of the things I noticed you're using the DMA in terms of your equipment. We have the, I noticed you mentioned the cold vapor system also in terms of some, which you did some, I started in red right at the top of the slide. So I'm wondering, we don't have the DMA, and that can be quite expensive to get into, but we do have the cold vapor facilities. How do we, and you're trying to develop a standard method, how would your method correspond to our method, and what can we do so that we could get some kind of, you could say, generally acceptable levels of congruence with our results, say from the cold vapor against your DMA, and what would we need to do, because it will be important that we put something like that in place to develop later on for these kind of international collaborations where you're using different techniques in terms of instrumentation. Thank you.
Thank you for the question. When you have a cold vapor, as long as you generate gaseous mercury and you can analyze it with a spectrometer, either fluorescences or absorption. So I guess you have absorption here, correct? So what is possible to do is to, for the solid matrices, I mean, you can mineralize it. The same as I did for the suspended particulate matter with reverse aquaregia. You can do it also for fish, for sediment, for soil. And then you have to dilute your sample to reduce the matrix effect and to have an acceptable level of aquaregia in your matrix. In our case, we work with 0.5% when we do cold vapor analysis with IFS after it, just because the gold trap is not supporting a lot of acid. Otherwise, you will destroy it and you won't be able to have a good analysis after. But then it's, as long as you continue to use certified reference material during your mineralization and you check the yield of mineralization with certified materials, and then you can do liquid analysis with sediment and soil and fish. It's just that we have a DMA and I'm lazy, and it's much more easier to do it on a DMA than doing it with a, yeah, we have 3, yes, true. But it's easier because you just have to crush the sample and you do the analysis. But we collaborate also with people around the world, in Brazil, for instance, and they do liquid analysis for fish sediments. It's just a matter of, using certified reference material to control the rate, the yield of mineralization, and then you can do coal vapor analysis for sure. Yeah.
Good morning, Alex from the Guyana Wildlife Commission. I have a question on the sampling, on the techniques. I noticed fishes would have been mentioned throughout the three presentations. What about other species of animals that utilize these waterways that you sample, like the capybaras, we have snakes, we have reptiles like the caimans. Is there any sampling of those species for mercury?
Yeah, thank you for your question. In French Guyana and in Bolivia and in Ecuador, we also sampled caimans because caimans, of course, sometimes are also eaten by local people. So we were worried about the mercury concentration in these animals, as they are fish eaters. So what we observed is that the mercury exposure of caimans in the in Amazonian rivers is at the same level than carnivorous fish species, carnivorous and piscivorous, but many carnivorous fish species. So the mercury exposure of caimans are quite high. So we also share the results with the local people and say that be careful by eating caimans. It can be, you have to take care, mainly for pregnant women and kids and children. Regarding terrestrial animals, I have colleagues, not me personally, but colleagues also sampled and analyzed terrestrial animals, and they didn't find exposure of high mercury concentration in terrestrial animals in Guyana.
There is also teams that sample fishing birds, for instance. I don't know exactly the result because we are not. Yeah, kingfish. Yeah, oh yeah, thank you. Kingfisher, because they eat fish, so it could be like for human people eating fish. But I won't say anything about the result because I'm not a specialist and I don't know exactly how much exposed they are or if there is any impact on their health. But kingfisher are also sampled in Peru and in Brazil. There is studies about that. But we are not really, with Laurence, for instance, in our lab, we are not really specialists of that, so we don't sample this kind of.
We have David Poix, il a fait ça. Yeah, we have a colleague who did mercury analysis in marine seabirds, in seabirds, and mainly all over the world in the Pacific, from the Arctic to the southern Pacific. And he also analyzed the mercury stable isotopes and to see the difference between the living places of these birds. And so you have some results, and of course they're high exposed, of course. But it was mainly to understand the mercury cycle between terrestrial and marine environments all over the world than the health issue, because people don't eat this kind of birds.
Any questions online? Okay. So maybe I'll just throw one out for, I guess it's applicable to all three presenters. In relation to tracing mercury in humans and the sources and how the the amount of mercury may vary based on diet. I'm wondering what are the methods you would use with regards to that? Because I can't remember, maybe I missed it, Loic, but did you talk about any complementary data that's collected when taking a human sample in relation to their diet or other habits? If you did, I missed it, and if you could just repeat it. If you didn't, you could just shine some light on that. And I wonder, in relation to what Alfred was talking about, whether or not this is the stable isotopes is the kind of tissues that you need to study stable isotopes. Can we take that ethically from human beings? So we're able to do the same kind of tracking of the sources? Because I presume you'd have to be doing self-reporting of the diet, not a dorsal muscle. But I'm just wondering about those things. Thanks.
Yeah, thank you, Calvin, for your question. So as I presented very, very quickly, we measured stable mercury isotopes in human samples, but only in human hair. Yeah. So as I explained in the presentation, human hair is the best biomarker for organic mercury exposure, for the methyl mercury exposure, which main source is the diet. So we analyze mercury stable isotopes in human hair, but also in the fishes, in the different fish species that people could also eat regularly. And what we observe is that it's really interesting because the advantage of the mercury stable isotope is that we have two types. I didn't have the time to explain, but we have two different fractionations. We have a mass-dependent fractionation, which is the small delta 202, and we have an independent mass fractionation. And very few elements, very few chemical elements, there's oxygen, sulfur, and so on. There are very few elements that present this double fractionation. So we use both fractionations, mass-dependent and mass-independent fractionation, to trace processes and to trace the sources. The mass-dependent fractionation is used to trace the processes of speciation of the mercury between the different compartments, between water, sediment, fish, human, so on. And we use the mass-independent fractionation ratio, which is the capital delta, 201 or 199, to identify and to trace the mercury sources in one sample. So for human hair, we see that we have a mass-dependent fractionation between prey and predator, for example, between fish and human. We have an inclusion, and we were the first to publish these results when we were working in Bolivia. So there is a mass-dependent fractionation between fish and human hair of 2 per mil. Yeah, and with this mass-dependent fractionation, we can see that there is a difference between prey and predator. We also analyze the stable mercury isotopes in human hair of pregnant women and of their kids, and we also observe like a predator, like prey predator between babies and women. It was really interesting. And we use the independent mass fractionation between fish and human hair to quantify and not to quantify, but to trace the different sources in the diet. And we see a difference in this mass-independent fractionation between freshwater fish species and marine water fish species, as I presented before. And we also see a difference between freshwater fish species that are living in the mainstream and freshwater fish species that are living also in floodplain lakes. We have a fractionation. So regarding the mass-independent fractionation, we can say which kind of fish species people are eating, marine or freshwater, and if these fish are mainly spending their lives in the mainstream or in the lakes. We published several papers on that if you're interested. But it's really a very interesting tool. Of course, we can't say nothing about the AGM impact on the human exposure. It's just only on the diet.
Thank you, panelists. We have come to the end of this fourth session. Thank you. And so now we will move on to the next session, and with my colleague from the Department of Chemistry, Ms. Lachmansingh. She will be the moderator moderator in this new session.
Good morning, everyone. Again, it's almost midday. I hope you all are refreshed after the snack. Yes? Good? All right. So I have the distinct pleasure of welcoming you to session 2, which is findings. So session 2 is actually a really long session. We have 10 presenters. So that means y'all getting lunch till 2 o'clock. I'm kidding. We will actually take 3 presentations first, and then we'll take a break for lunch, and then we'll come back to the last 7. So I'm really happy and excited about the finding session. The finding session, we're first of all going to hear from our international partner and their research and some of the findings they've basically accumulated with their work in Guyana. But also, we're going to hear from a lot of work that was done by our local people from the University of Guyana, students and staff. Some of these presentations were undergraduate projects, people from chemistry, biology, environmental sciences. And so from a wide range of disciplines, we're able to collate this session now for you on the various topics related to mercury, mining in Guyana, impacts of gold mining. So it's, you're going to hear about impacts on human health. We did a few studies on mercury found in hair samples of people in indigenous villages. We're going to hear about biodiversity impacts as well, forest cover, and then also I know one of our colleagues, or 2 of them, are going to be more on the social sciences side, where they're going to be looking at how has it impacted our achievement of sustainable development goals, or our progress to it anyways. So looking forward to a really exciting session and our ability at the University of Guyana to showcase what we are capable of doing with very little or limited resources, and to make sure that our international partners see the value or the potential in our people and can help to support us in the future. So let me start by introducing or welcoming Dr. Loic Martin from IRD, and then we'll go into the other presenters, Rochelle and Benito.
Yeah, it's me again. Don't worry, it's my last talk. So I'm gonna present all the findings we have with the mercury phase-out project in Guyana. So, before to start, I will thank my colleagues from Toulouse, Jeroen and Laurence and Laure, for all the work in the lab and in administration and so on. And also, I cannot forget Cleveland and Aiesha for the Guyanese part of the work and all the help to make the field possible and all the mission possible, because organizing that from France was quite hard. So it was, yeah, without both of them, nothing of those results would have been possible to be shown today. And also the people who helped us and came with us on the field, Rochelle, who is here, she's not listening, but yeah. And also Elford and Sion, who is not there today, but without them also it would have been much more difficult to sample all the river in Guyana. So the supporting mercury phase-out project in the Guianas was done both in Suriname and Guyana. I will just focus on the Guyanese part today. With Laurence, we are working on the component 3, which is the regional mercury observatory and the evidence-based mercury phase-out. So we raised 2 questions when we start the project. What is the spread and the origin of mercury contamination in the different regions in Guyana? And also, what is the level of exposure of the riparian population? So we focused on aquatic environment and aquatic systems. So the mercury cycle in those systems has two kinds of sources. Natural sources coming from natural erosion and soil mobilization. So it's called the geogenic mercury. And we also have some anthropogenic sources. The first one comes from artisanal and small-scale gold mining, which is amalgam burning. By burning the amalgam, you release gaseous mercury in the atmosphere, and this gaseous mercury is deposited in the area. And this is a source of mercury contamination for soil, and after erosion, it's a possible contamination for river systems. We also have when they do the process of recovering the gold, they use liquid mercury before burning amalgamation, and you can have also release of liquid mercury in the environment. And the last is deforestation, also due to gold mining, because you need to deforest when you do gold mining, but also for slash and burn agriculture or logging, for instance. So when you deforest, you have an excess, you create an excess of erosion that release a lot of particles in the river. So all of those sources play in the mercury mobilization to rivers, and in those rivers you have methylation, and this is a conversion of inorganic mercury to organic mercury, and this organic mercury form is the one that is accumulated and bio-amplified in the food chain, in fish, and that could threat the health of human people living along those rivers. So the area we studied in Guyana were quite, the idea was to have pristine areas and mining areas. Pristine was quite tough to find, actually. We tried to work a bit in Iwo-Krama, in the Buro River, or in the Siparoni rivers, on their tributaries. In Iwo-Krama, you can have some pristine rivers, let's say like that. And we work also in the Potaro River basin and the Mazaruni River basin. Mostly around Madia and Maikobi in the Potaro River, and to the mixing with the Shekibo River. And in the Mazaruni, it was mostly the lower and the middle Mazaruni from Isineru to Bartika. And all the tributaries, not all the tributaries, the one we could access. We did two sampling campaigns for this project. We wanted to have one in low flow, one in high flow, but it was not really possible to do that. So the hydrological conditions were roughly the same, but sometimes the river was a bit lower than expected. But let's say that it was mostly high flow compared to the low flow you have today, for instance. The sample we took were water, fresh water, sediments, soil, and tailings. The biological sample were human hair and fish, of course. And for human hair sampling, we worked with mostly with the indigenous village, Isineru, Kangaruma, and Tasirin in the middle Mazaruni. Fairview when we were in Iwo-Krama, and Maikobi and Campbellton Madia when we were in the Potaro area. First, I will present the environmental data. Mercury in river. So we measured two kinds of mercury. We did total mercury analysis, and speciation analysis. Total mercury by gold trap CVIFS, like I presented earlier, on the dissolved colloidal fraction, which is below 0.45 micrometer, and also on the suspended particulate matter. And for the dissolved, we did also an analysis in organic mercury with Persian trap GC CVIFS to have the methyl mercury fraction of this water. So the results, basically, all the organic mercury concentration were below our detection limits. We had some issue, to be honest, with the device. So the detection limit is a bit higher than we expected, so that's why we were not able to really have concentration in methyl mercury in this water. But anyway, the fraction is really low. 0.1 nanogram per liter is really a low value in terms of mercury concentration in water. And to give you an idea, I forgot to mention it, but No, maybe after it. I don't know. Why I cannot move again? Ah, okay. Okay, so sorry. So yeah, the drinking water limit is 2,000 nanogram per liter from the WHO and US EPA. So it means that basically there is nothing when we speak about 0.1 nanogram per liter. If we go to the different area where we sample, so we see that here on the top it's dissolved fraction in terms of mercury, total mercury concentration, and here is the suspended particulate matter. We range from 1 nanogram per liter to the highest one was around 30 nanogram per liter in the dissolved. It was observed in the Potaro area. And in the suspended particulate matter, it's the same. We range from 1 to 50 nanogram per liter, which is quite low in terms of mercury concentration in the water column. There is no significant difference between mining areas and non-mining areas. So the level of mercury in waters is basically the same everywhere, with some differences. We see that here it's, so in green, no, not in green, in brown, it's the Essequibo. In blue, it's the tributary of the Mazarunis. In dark blue, it's the Mazaruni itself. Here in gold, are the tributaries of the Potaro River, and the Potaro River here, and in green, it's the Siparuni area. So we see that maybe the Potaro is a bit higher than the others. We can explain that by a lower pH, but we didn't see any correlation between total mercury concentration and pH, or no correlation also between total mercury concentration in the water and the organic carbon concentration in the water, which are the chemical physical parameters that play the most for the mercury speciation and for the mercury in the dissolved phase of the water. Now, total mercury in soil, sediment, and tailings. So here are sediment in mining areas, here in pristine areas for the Mazaruni. Here is the tailings, and here are sediment in mining areas for all the rivers, Potaro, Mazaruni, and Siparuni. Sediment in pristine areas, tailings, so mining waste, basically, everything that we recovered from mining activities. Soil in pristine areas, and soil in mining areas. All was analyzed by a direct mercury analyzer, and we didn't see any differences between the different compartments. The concentrations were quite homogeneous, and there is no higher concentration of mercury in tailings, for instance, and there is not so much difference also between mining areas and pristine areas. Just to compare a bit, those are data from French Guyana. Well, the screen is low, so we cannot see it really well, but we are in an average range of 100 to 150 ppb of mercury in the solid phases in Guyana. And in French Guyana, depending on the area, it was a bit higher, but basically we are in the same range of what we observe in French Guyana, which is quite logical at the end because we are in the same geological unit. Yeah, so now comes the funny part. So, so we did, for the sediment and the soil and the tailings, mercury stable isotope analysis. Basically we digest the sample in aquaregia, we do purge and trap to purify the matrix, and then we do analysis with cold vapor to generate gaseous mercury, and we couple that to a mass counting ICP-MS, meaning that with that we are able to count the abundance of the mercury isotopes. So what you see in this graph is, so as Laurence explained a bit during the question, we have two kinds of fractionation in mercury. A mass-dependent one, which is on this axis, small delta 202, and an independent mass fractionation which is big delta 9Y1. So all the dots you see here are the result for Guyana. We have square, which are the soil. The diamond are the river sediments. The cross here are for liquid mercury, and the triangle are for tailings, or mining waste in general. When it's in green, it means that it's pristine. When we say pristine or mining area, it's based on the local knowledge or what we see on a map. A pristine river is not mined, supposedly, and mining area is a river where there is mining activities that were done either in the past or are active. And we also have black dots. You cannot see them really well, but it's what we sample downstream mining activities around Bartica between the mixing of the Kuyuni and the Mazaruni and the Essequibo at this area. You have also some tailings here. The two green, I don't know if you see well, are those from French Guyana. They are really high in concentration. We know that there is liquid mercury in it. And we also have those one from Guyana, that one is here and one is here. I will come back to that later. What we see also is that there is it's a mixing model. There is two end members. One is due to ore and mine tailings, associated also with liquid mercury, and also anthropogenic gaseous mercury emission, which means that all of that is one end member. And we have also pristine areas, and to this end member, I mean, you have you are going close to zero in the big delta anomaly. In this side, you have the pristine sediment and the pristine soil, or undisturbed soil. Those ones, they are lower in, more negative in both big delta and small delta fractionation. So what we see in the Guyanese river, basically, is that there is a mixing between ore, mine tailings, and liquid mercury in sediment with undisturbed and pristine sediments. And some rivers are more impacted than others by this kind of mixing. A low anomaly, an anomaly, a big delta anomaly close to zero doesn't mean necessarily liquid mercury. It could be a mixing with an ore or a mine tailing that is not full of liquid mercury. The concentration will help us to understand, the concentration in mercury, I mean, will help us to understand if there is liquid mercury or not. In the case of Guyana, when we check our results, it comes to the second graph. So I put on this area the big delta. Yeah, I know. Okay, so, no, next slide, yeah, thanks. No, yeah. What you see here is that, so, there is no relationship between mercury concentration and the big delta anomaly, meaning that there is no increase of, there is no pollution of liquid mercury at regional scale. We cannot find it in the sediment, because you see an increase, a pollution in liquid mercury means an addition of mercury based on the geological background that you will find in sediment. In this case, the liquid mercury input could be summarized by an anomaly closing to zero. And all those group of samples, for instance here, they are closing to zero. But the concentration is quite low. It's close to the geogenic background, which is around 100 ppb of mercury. So what we see is that the geogenic background remains dominant in our system, but it doesn't mean, it means that there is I think no liquid, no liquid input of, no liquid mercury input in the system. But it doesn't mean that there is no impact. It means that the impact is different. If we think in terms of mercury fluxes, for instance, it will be really more interesting because by gold mining activities, we generate more erosion and we release a lot of sediment in the system. And if you observe a reserve in Guyana, a river in Guyana, you will see that all the sedimental dynamics in a river is completely disturbed. There is sediment transport that occurs in an area where it's not supposed to occur. So this will generate fluxes of mercury that are really high, and we don't know what are the consequences of that. And this is not only thinking of input of liquid mercury is important because we can have a huge amount of, big fluxes of mercury that are not really seen by those kind of tools. And this is quite important to think like that for the river system here in Guyana because the sedimental dynamics is completely disturbed in rivers. So just to finish, I will be quick on biological data. So it changed completely. Now we measure mercury in human air. So human air are a biomarker of organic mercury exposition. So basically, it's due to diet, fish diet mostly, and the WHO limit is 10 ppm. But over 5, we say that it starts to be harmful for human health. In the villages, we assess to see that Madia Campbellton is the area where there is less exposure to mercury, and we see that few people are over 10 ppm, and most of them, they are below 5. When we go to Microbe, it starts to increase, and we see that the proportion of people above 10, above 5, it's the main proportion. And it's like that for Tarsirini, Kangaruma, Isineru, and Fairview. And what it means is that the further you live from a town, the more you rely on fresh water fish for your diet. So Kangaruma is the one with, for instance, with the highest level of mercury in the population, and is the one which is the most remote. And the people there rely a lot on fish. This is not linked to gold mining activities. It's just a question of how you can diversify your food, how you have access to a diversified diet, and how much you rely on fish to eat. So the conclusion is that water are quite clean in Guyana in terms of mercury, low level, below the drinking level that is recommended by WHO. There is no significant difference between pristine and unpristine areas in terms of mercury concentration in sediments, but also in water. No liquid pollution at regional river scale. And the level of the level of exposure to organic mercury is high as long as you live far away from a town. So thank you for your attention. And sorry for the delay.
All right, thanks, Dr. Loic. So the next set of presenters, I just want to warn that we have 10 minutes each. And I will give you a little warning when you have 2 minutes left. We're just trying to keep everything tight in terms of time. So up next we have Ms. Rochelle Bino, and then we'll have Benita Davis right after that.
I hope you can see me. I'm a bit short.
Yes.
Okay, good day everyone. My name is Rochelle Bino, BSc Chemistry, and well, good day to you all. Well, my project that I did is based on mercury contamination, particularly in the Potaro mining district.
Now, first of all, the reason why I chose to do this experiment. It has been known over the years that there has been an increase in heavy metals being released into the environment, and this is due to mining activities. One of those metals predominantly, which we're all here to investigate, is mercury. Now, we all know the description of mercury. The one we're used to is the metallic form of mercury, which is elemental mercury, we refer to as quicksilver. However, mercury can be in its organic form and its inorganic form. The organic form is the methyl form, which is most dangerous and most toxic, and the one we are most interested in right now. So, people in the Amerindian community have been exposed, their livelihoods have been put at risk because of mercury exposure. They use running water for carrying out basic livelihoods, and this water has been put them at risk. Elevated levels of methyl mercury can cause irreversible damage, such as the reduction of their motor capacity, harm to developing fetuses, and breastfeeding children. From all of the research that we've heard so far, there has been ongoing research from people right here, WWF, IRD, and GGMC, and other organizations, which have already proved that there has been high levels of mercury contamination, particularly in hair and fishes, large fishes. Only in 2022, samples were brought back to Region 8 with mercury levels in here that are higher than the prescribed levels, most of them being above 1 ppm, which it should be below, some having even up to 20 ppm of mercury. Now, it is known that artisanal and small-scale gold mining accounts for about 70% of the gold produced in Guyana. Now, if there's a rise in the gold output, there's obviously going to be a rise in the amount of mercury being released, since we use mercury to extract. Now, what is my objective based on this information? Since I'm investigating water, I want to determine the concentration of mercury in my river water samples collected in Region 8. I want to determine the concentration of mercury in some rainwater samples, and I also wanted to determine if there's any significant variation between the rainwater samples and river water samples collected in these locations. Methodology. Now, my project was supported by WWF and IRD with the help of Dr. Lewick and Mr. Cleavon Cameron. The study area here was done mostly around in this cluster, surrounded around Madia. The areas where rainwater samples were taken from were directly in Madia, and a rainwater sample was also taken from University of Guyana campus for comparison to determine if there was any much difference. The river water samples were taken from along the Potaro River at various points using coordinates, and I structured them from downstream to upstream to determine the difference. This is just an aerial view of the White Hole mine where one of the water samples were retrieved from. Now, the stages of mercury analysis. I can't get into all the details of exactly how the scientific research was done, but samples were prepared. Sampling was done. I told you the areas. They were stored. The samples were digested. I prepared my standards and blanks to run it via Atlant Model Vapor Generation Accessory, AAS, Co-Vapor AAS. Now, one thing to note about the sampling is that low-level mercury samples are very susceptible to contamination. And for this reason, just like Dr. Lewick would have explained, we use PTFE bottles and we acidify them to a pH which is less than 2. I think it was 0.04%, so that to minimize the loss of mercury. I put my results in a table. We had instrument concentrations, however, from my calculations, we would have had the actual concentration, for which the rainwater samples range from 0.03 to 0.19 micrograms per liter, and river water samples from 0.48 to 0.93 micrograms per liter. That is ppm. Now, what we should know is that the maximum contaminant level of mercury that is stipulated by the EPA is 2 ppb, or 2 micrograms per liter. The accepted range, however, is 6 micrograms per liter
by WHO, while the range for EU is below 1 microgram per liter. For my experiment, I used EPA's maximum contaminant level. And I did a simple bar graph, which is easier to understand, where the blue line represents the maximum contaminant level. Now, from this table, we can see that all of my sample results fell way below the range of the maximum contaminant level for mercury. However, it should be noted that detectable levels of mercury are hard to find in running water. So from the time you can find detectable levels of mercury, it shows some amount of contamination. However, it is nothing to be scared about. So we can also notice a trend where downstream has higher levels of mercury contamination than upstream, and there will be a reason for this. So I put it into a simple line graph to show that there is downward progression, meaning that there are higher levels of mercury downstream. The reason for this, a lot of the mining is done upstream. However, as you go downstream, there is more accumulation of mercury or deposition. So as you notice in the middle, on the second sample, which was taken via coordinates, it has slightly higher levels than our upstream sample. That's because as mercury and water flows down, a lot of it would have accumulated in sediments, and downstream has more swampy areas, more sediments, etc. Now here are the rainwater samples. They were taken from White Hole, which is directly in one of the largest mines there, Hines Farm, and on the University of Guyana campus. So we can see that the campus has the lowest level of mercury because in those areas there are higher levels of mercury. Hines Farm tends to have more mercury than White Hole, which might not have been expected while doing the experiment. However, this is due to throughfall, and that is the fact that is due to the water cycle. So a lot of the mercury in the atmosphere, which is released from burning amalgam, etc., would have been accumulated on leaves, plants, etc. When the rain falls, a lot of those mercury would have been picked up in the droplets of water, which come in their rainwater samples. So we see a higher level of mercury in the farm than in the mine itself. So one of the things I wanted to determine is if there were any significant variations between the rainwater samples and the river water samples. From the results, we can see clear variation. However, statistical analysis was done to prove this. These are the results of the statistical analysis, having a mean of 0.68, observed mean difference of 0.53, and a probability of 0.035. Now, my hypothesis was that there is significant variation in the mean concentrations of mercury samples collected from river water samples and collected from rainwater samples. The significance level was 0.05, and because my probability was less than that significance level, I can reject the null hypothesis and prove that there is significant variation. One of the limitations I had was that there was a limited amount of sample obtained, which I would have preferred to have more amounts of samples to be able to compare, but this is due to shared research and limited sampling storage material at the time. So a smaller sample size can give you a greater margin of error. However, the results are quite cohesive. So some of the things I recommend, which some of them are probably already implemented, would be routine and yearly checks to monitor the levels of mercury, because the levels are low enough right now in the running surface waters to ensure safety, to prevent a drastic rise in the levels of mercury, to be able to alert the indigenous communities of when the waters are not safe for their usage, granting the indigenous communities full protection rights against their waters, and developing, most of all, total maximum daily loads, which are the calculations of maximum amounts of specific pollutants that a water body can receive and still safely meet the water quality standards. That has brought me to the end of my presentation, showing that all rainwater samples and river water samples that have been tested by myself with the help of Mr. Lewick and Mr. Cleavon fall below the maximum contaminant level and can still be used by the indigenous community.
Well, all protocols observed, a pleasant good day to you all. I'm Benita Davis, and my task this afternoon, I think it's afternoon, is to present on my final year research. Which is entitled An Examination of Physical-Chemical Water Quality Parameters for Rivers Within the Vicinity of Kaieteur National Park. And this was done in collaboration with Protected Areas Commission and GGMC. Okay, so why Kaieteur National Park? So Kaieteur National Park is the home to the largest single drop waterfall in the world. It's approximately 741 feet, and it's also a protected area because of its high levels of endemism, primarily the fish population of the upper Patara River. Unfortunately, in 2016 and 2017, we noticed illegal gold mining activities occurring within the buffer zone of the park. So GTMC and the PAC came together and said that, they wanted research to be done to really set up a baseline because, like the slide shows, there is no comprehensive baseline water quality data for Kaieteur National Park. So this research aims to set up that baseline so that future research can be compared to what was established here. Objectives. My main objectives are to determine the physical-chemical parameters for water quality for 4 areas along the Ichiraq, Muri, and Patara River, and then to determine the concentration of mercury in water and sediment samples for Patara and Ichiraq River, and then finally to investigate what relationship exists between those physical-chemical parameters as well as the total mercury concentration. So now this is a map of the sample site, so the area in green. Sorry. This is not my slide. Yeah. So the area in green here shows the protected areas commission. So this is Kaieteur National Park, and this is Kaieteur National Park. The area in yellow are the mining blocks. Okay, thank you. Are the mining blocks, and then we'll see the sample points. Yes, so the area in green, Kaieteur National Park, here's the Patara River, this is the Kaieteur Falls, and the green dots that you see along are the sample points. The yellow blocks are the mining camps that existed. And so Ichiraq was measured, and you see, well, in 2018, there were 4 active mining claims at the time. I'm not sure the state of the river now. And this is an indigenous community of Chenapau that is nearby that river. And this is just a clear visualization of my sample points. Before, after the fall on, this is the Muri River, and the other one is the Ichiraq River. Having a tough time. So the method used for water samples. First of all, the HAC multi-parameter meter was used to measure the water samples in situ, so 6 parameters were analyzed. Then a depth sampler was used to remove said water, placed into a plastic bottle, PTFE Teflon, acidified by 2 mL of nitric acid, stored at 4 degrees Celsius, and then transported to the lab where it was analyzed using EPA method 1431. 1631, sorry. And then the method used for sediment samples. So an Ekman grab was used to collect the sediment samples, and the uppermost 4 the uppermost. Clicking this, right? I'm clicking to the no. Yeah, so sorry about that. So the method used for sediment samples, the Ekman grab, and then the upper 4 to 5 cm was removed, placed into a bowl, and then the first 2 steps repeated to form a composite sample. Then that sample was subdivided into 3 parcels and placed into Ziploc bags and into a cooler with ice at 4 degrees Celsius and transported to the lab. Where EPA methods 3050B and 1631 were used to analyze it. Okay. Results and discussion. Well, I really can't do the results and discussion without the graph. But what we found is that turbidity was quite high. So although 6 parameters were analyzed, the turbidity and pH were well above the international guidelines set by WHO as well as US EPA. And this confirmed with studies done in Eastern Cameroon as well as Suriname. However, they attributed their high turbidity values to artisanal gold mining. But in this case, the areas that had the highest turbidity were found in locations that were much farther away from the gold mining activities. So it's more plausible to conclude that this high turbidity value is more linked to the bands of mudstone in the riverbed, so suspended particles, because as the river rolls over the clay, it suspends the particle, increasing turbidity. And this is black water we're talking about. So the tannins present, which gives it that tea color, yes, that present gives it that tea color, increases turbidity as well. In terms of pH, okay, yeah. So in terms of pH, which is this graph here, we see that they were generally acidic, which is natural for black water rivers because of the tannic acid present. However, they were said to be high in pH simply because of the geological formation as well. So the rocks that make up the Kaieteur National Park are acidic rocks. So they're granite, quartz, and mudstone. So the water flowing over them might also result in this acidic solution that was found. Now, objective 2, total mercury in water. All of the sample sites were well below international guidelines. However, Ichirok, which had the 4 mining claims in 2018, were higher than Pataro. I cannot say that that is because of the mining claims, simply because there's no baseline. I cannot compare it to anything. However, the values found were similar to Narti et al., who conducted studies in the North District of Ghana. And what they found, or they attributed the low levels of mercury to, is the natural occurrence of mercury in that area. So it could be the case. Objective 2. Yes, total mercury in sediment. This was also well below the international guideline set. And for our site, which is Kaieteur National Park, that you had the illegal mining, you also have mining camps outside of this protected area. It was well below areas such as Conishan, as well as Irakram, or other protected areas in Guyana that were studied in 2010 and said to be pristine at that point in time. So Kaieteur National Park was not influenced that much by gold mining or mercury levels, and it was also very pristine. Now, total mercury in water versus turbidity. Based on this graph, you see a similar trend exists between total mercury as well as turbidity. So we can say that there was a direct or positive relationship between the turbidity of the water as well as the total mercury concentration. So the higher the turbidity, the higher the total mercury concentration, which confirms the study done by Ubota et al. In Cameroon. And now here, objective 3, we compare total mercury for both Ichirok and Pataro. So with the exception of 0.4, they were generally the same. So based on observations, it was noted that the sediments of 0.1, 2, 3, and 5 were sand particles, while for 0.4, it was clay particles. So we can clearly state or conclude that there's an inverse relationship between particle size and total mercury concentration. So the finer the particles, the higher the total mercury. And so in conclusion, all physicochemical parameters were analyzed. They were found to be safe based on WHO guidelines with the exception of pH and turbidity. For total mercury in water as well as sediment, it was well below international guidelines and so safe for both aquatic and human environment. And finally, only a weak positive correlation existed between turbidity and total mercury in water. And then in terms of mercury in sediment, there was an inverse relationship with particle size. I do recommend that future research is conducted to have seasonal comparisons. For this, research was done in the dry season, so there needs to be research done in the water season, or the wet season, so we can really see the changes in concentrations. There also needs to be research done in hair and tissue samples, hair and fish, samples, so that we can see how it impacts the human life, because there is a community of Chinapa that is nearby this region. And finally, I hope that a baseline water quality data can be set up using this research so that future research can be used to properly monitor the Kaieteur National Park. Thank you very much.
Okay, can I have the three presenters on stage as well again? Thank you. So we'll have a short question and answer segment. And I want to note that Ms. Bino's work, her mercury analysis was done right here at the University of Guyana. She only did it this year. So just to know that we have the capacity to do it at UG. All right, so yes, we'll take some questions from the audience.
Good morning. Johan Waldron from the Guyana Wildlife Conservation Management Commission. Congratulations on the presentations, they were well done. And I have a question for Ms. Bino. As part of the recommendations, I'm just wondering if cyanide was considered as a potential replacement or substitute for mercury in these areas to potentially help reduce the concentrations and contamination levels that you encountered.
Okay, so by myself, definitely cyanide was not considered, but based on the research that I've done, I read a few and nobody mentioned cyanide as being considered. And I think one of the reasons is because cyanide is also quite dangerous. So there are many risks that will come up in terms of using cyanide. And plus, mercury right now is one of the most effective and most affordable methods being used for small-scale gold mining. So it would be difficult to change.
So cyanide is used in the larger scale mines, but as Rochelle said, for the small scale miners, a bit of an adaptability for them. And then it's a lot of, you need a chemist probably on site. So I don't know if, yeah, I know some of our colleagues actually we have sitting in our audience there, Deshaun, and yeah, we'll be able to speak to some of that as well. So.
Got you.
Yes, so my name is Shamisa Tom. I'm a mineral processing engineer from Guyana Geology and Mines Commission. Cyanide is permitted for use by large-scale miners, but not by small-scale miners. Basically because small-scale miners, it would require more technical personnel for them to store and use the cyanide. Also, we have social problems with cyanide as a country, and we should know that as Guyanese. We had the Jim Jones story, and we also have persons, we don't want persons to use it as a poison, so to speak, socially to harm others. There's also a consideration, or even themselves, with suicide in Guyana. So cyanide requires a permit and is used by large-scale miners. Cyanide is also, I heard my colleague says it's dangerous, but it's a preferable method to mercury because cyanide can be broken down into its components, because it's a compound. But mercury cannot be further broken down into any other thing because it's just an element. So that makes it a bit more environmentally friendly to be used than mercury, right? Okay, that's about it.
If I may add, there is cyanidation in artisanal small-scale mining. It's a technique that is used in Western Africa, in Burkina Faso and Niger, for instance, and also in Suriname. In Suriname, in the Brokopondo area, and also in Maroni River, there is artisanal small-scale miners who use cyanidation as a technique. Basically, they take the waste from the mercury process and they use cyanidation to recover the 50% that remain in the mining waste to get the remaining gold. So in Suriname, it's a technique that is widely used now in artisanal and small-scale mining. So.
Just to add to that, cyanide can also be broken down by sunlight, so that also adds to the environmental friendly part of it.
William Wolford, representing the Miners Association. I just want to put some information out here because we just talked about the Potaro Basin and we talked about Madi early, we talked about Mykobi early, and we talked about it in relation to the small-scale miners. But historically, a huge set of large-scale operations were conducted in the Madi River and over in the watershed of the other part, the Minnehaha River, and then down further, the Conourok River from source all the way down to Long Falls. And in Chumachimari area on the Potaro River, huge dredges were operating for dozens of years. And it's interesting that all of that took place, and I have never found any significant information that said that residents and so on were badly affected by mercury poisoning. I'm still scratching my head on it because the Madia River itself, when you go to Madia, there's a Madia River, and there was a dredge above the falls. There's a small falls in Madia River and below the falls. And when we did work there as Georgia Mines Commission, looking at the outflow from the Madia River into the Potaro, you hardly found anything. So I think we should focus on studies on what's happening. Is that mercury? And in the 1980s, I was examining a dredge operation, a local dredge operation in the Potaro River, about 4 miles down from Mykobi, and the guys pulled up, and in the sluice box there was a globule of amalgam gold with mercury in it. They didn't put it there, they pulled it up from the Potaro riverbed. I saw it. So I'm interested in some studies on is there something going on where the mercury is amalgamated and some place below there and it's not moving or whatever it is. But the major work with mercury in the Potaro area and so on was done by large-scale bucket line dredges that operated for dozens of years. The last one operated in 1959, right by the Chumachimari Falls, where they have that hydroelectric plant. So I would ask that we recognize small-scale guys doing a little mercury, but try to figure out why we haven't had any systematic research done as to why you have big bucket line dredges using mercury in the sluice box, operated for dozens of years, and we can't seem to figure out where the mercury went. And this other thing is that one of the presenters mentioned the fact that the geology of the area makes a difference as well. What is happening in the Potaro area, along Madia, along Conourok, is that there's a set of material called quartz pebble conglomerates. It's the sandstones and conglomerates. Those quartz pebble conglomerates are known internationally, and Guyana is classified, Brazil, Johannesburg area, and in Elliott Lake, Ontario. They're known to carry certain things, including gold and uranium, but one of the trace metals that they've always identified is mercury. So you would expect that the quartz pebble conglomerates, that Potaro River, Conourok River, and so on, would be giving out something as it breaks down and so on. So I'm interested in further studies done in these areas. Thank you very much.
Samisa Tom again from GGMC. I must commend the presenters for the work they did. I really commend them for the results also that were brought forth here today. Also, I'd like to see the university, as Mr. Wolford rightly said, doing work on where the mercury goes, because mercury is not soluble in water. So something happens to the mercury. And Ms. Davis rightly mentioned that in her studies, where there were finer particles, she found more mercury there because of the, I guess because of the suspended solids that would be where finer particles exist more than where a sandy sample would be. There would be less suspended solids, but that does not necessarily go to say that that area has less mercury than a siltier clay area. So we need to study where the mercury goes and probably do sampling on riverbeds or something of the sort. Thank you.
So, just to jump on the two comments, it's the mercury cycle is quite complex. When it's in mining areas, when you do mining and you use liquid mercury, it's mercury zero. It's on the liquid form. It's an heavy metal. So, of course, when it's on this form, it's heavy. It won't travel far away from the mining site because, you know, when you put a solid in the water, the easier it is, the quicker it will go down to the bottom of the glass, for instance. It's the same when you think of the sediment transport in a river. But even if there is liquid mercury in a sediment, deposited in a sediment, after some time it will come mercury 2. This one is a reactive form, and it can be trapped on carbon that is dissolved in the river. It could be trapped on fine particles which are really reactive. So then it can be transported further away from the mining site. What is tricky with mercury cycle in gold mining areas is that also we have, in Guyana, in the Guyanese shield, the rivers are quite low in sediment loads because the shield is old and there is no more weathering, no more erosion. So mining activities, they are creating a new dynamic by creating more erosion and new sediment transport. And sometimes, if you erode a soil, you have the geogenic signal of mercury. It's, let's say, natural mercury, but it will be transported, and sometimes it can, when we do a sampling, if you have 50 centimeters of soil eroded by mining activities, you won't catch what is below that, and you won't catch, for instance, old liquid mercury that could be accumulated there due to old dredges activities or so on. And it's really difficult also because sediment transport is a way of research by itself, and in the case of a river, when you have low and high flow, during low high flow, it's like in your toilet when you flush it, you know, you have some sediment accumulated in one area, and 1 year before it could be in another area because it has been flushed away by the water. So in terms of mercury cycle in mining areas, we need to think about the sediment fluxes and how it's evolved due to mining activities. And not finding liquid mercury doesn't mean that there is no I won't say pollution, because the pollution means that there is an increase of the geogenic background. But it doesn't mean that there is no more mercury fluxes or there is no impact on the river, because you change the sediment dynamics. You can increase the fluxes of mercury, because if you reject more sediments, okay, there is less mercury concentration in those sediments, but at the end there is a lot of sediment compared to what you will have in a natural way. So there is more mercury in terms of mass in your system, you know. So just looking at mercury concentration, sometimes it's not enough. And mining activities as I said before, they really increase and change the natural dynamic of a river, and this is damaging, and this also has a huge impact on the mercury cycle. And we cannot just think in terms of liquid mercury, because yes, it's harmful, it's dangerous, it's a pollutant, but what happens when you have a huge mass of mercury, natural mercury, coming in an area where there is flooding, for instance? In those areas, you will have a change of inorganic mercury to organic mercury. And as we presented during those talks, organic mercury is a dangerous form. Yeah, I finished. And this also, we don't know what happened. It's a human process, because we change the dynamic, but we can increase the rate of methyl mercury creation. And what happened to the food chain? This is the question that remains for mining activities.
Thank you. Just a quick one. I couldn't, being a member of the chemistry department, allow that statement to run with respect to the research. It's a good idea coming from Mr. Wilford as well as from the GGMC, right? And Ms. Stoughton, right? The whole idea now is that before we didn't have the capability to do this kind of analysis in-house. We now have it. So maybe we should also with WWF, and so we can look at those kind of things. And it's easy to set up projects for various students and work around and those kind of things. So it's open. Let us discuss it. Thank you.
Open for collaboration, and especially logistical assistance. We know we always get support, but yeah, definitely.
So I just wanted to flag on the same line that if you note, part of what we're trying to do in this is to come up with things that we may be able to collaborate on. What are the priorities? So I want to keep this same kind of thought as we go into other sessions, and particularly tomorrow morning when we'll have. So this is a plug to encourage everybody that's here to come back out tomorrow morning for us to have that discourse.
Yeah, and I think you might eventually see in some of the other presentations where the mercury ends up. So Loic gave you the very chemistry description, but in some of the presentations to come this afternoon, you're going to see where it actually ends up, or maybe in the fishes, or, you know. So that's, yeah, all in all though, I want to commend our presenters today for all of their really hard work. And all of the significant findings, and guys, keep it going. We can only make it better from here. So we're gonna take chair, half hour for lunch.